situation and what is going on in the world. It feels almost unreal that we are discussing science in such times. But on the other hand, science is exactly what we need in some way, something that unites us, something that is about optimism and the possibility for a better world. And we thought that maybe there are many initiatives going around, different institutes that are starting initiatives to help the scientists from Ukraine. So if you want to share an initiative that is going on in your institute in the chat, we'll try to put them together and post them later on so people will have it accessible in one format. This is our second year. The whole program started in the middle of what we thought also isn't possible, a pandemic that was crazy. But I think one of the good things that came out of that is this opportunity to bring people from all over the world, all over disciplines, to discuss this emerging need for something that is happening in science, that we're starting to look at science as an integrated biology. We had amazing speakers from all over the world, from psychology to worm biology, everything, as long as it tries to break the classical boundaries that we are confining ourselves with science. We had speakers from all levels of academic careers, prominent speakers like the one we are lucky and honored to have today, and the postdocs that were amazing and hopefully are already working as an independent PIs. So if you are in this stage of the career and interested in this stage, we are happy to give this opportunity. And of course, if you have new ideas for fields that you feel should be integrated here, we are more than happy to be part of that. This whole program is run basically, maybe let me just try to do the share part. I want to share this so you can see the people who are actually doing that. So this whole program is run by a wonderful group of postdocs and students, Raquel, Sam, Jerica, Zita, who is here, and Jerica will join us later, Mariam and Rita, and 3 new members, Sylvia, Lina, and Corina. And basically, Carlos and I, we are really just a support team. I have to say, not very reliable in most times, but we are there. And we are really grateful for the great audience that keeps coming back and asking questions that are really what this opportunity is here, that people from different fields asking unexpected questions. And with that, I will stop my share, which I don't know how to do, of course. Zita, can you help me? Thank you. And I will present our speaker for today. Ruslan, I think for many of us, really does not need an introduction. I think his discoveries has made a change in the way we perceive immunology with the discovery of toll-like receptors, which of course, I think changed the way we think of immunology. But I think mostly even his recent works that are really kind of breaking the boundaries. He's working in the biology of inflammation, mechanism of disease, and I think he's trying to think of this whole world of medicine in evolutionary perspective. And this brings something very different to the field. So more formally, Ruslan joined the faculty and Department of Immunology in Yale University in 1999. He is currently a Stern Professor of Immunology at Yale University, and he is a Harvard Hughes Medical Institute investigator. But I think mostly if I need to introduce Ruslan, I will say he's just one of the greatest thinkers that we have out there today, and we are very lucky to have you today and to join us. So we're looking forward to this presentation, and then we will have some more in detail discussion. So please, the stage is yours.
Well, thank you very much, Asya, for this really generous introduction. And thank you to all of you, Carlos and Zita, also for inviting me. It's a great pleasure to be here despite the difficult times. It's probably been the most disoriented I have been to prepare for a seminar. And of course, we all hope that there will be better times in the near future. So I gave this title when Asya asked me some months ago, and then when I was about to prepare, I started to think, what did I have in mind when I gave such a title? So, so what I thought I would do, I will try to bring up some questions. Mostly, it's not going to be a sort of a traditional type of presentation. It's mostly to bring up some questions that I feel might be good to discuss together, and I will try to speak about some possible, maybe not so obvious connections in the way that the immune and nervous system works, and some related questions that I just find very interesting, and I hope you will find them interesting too. So I will try to share the screen, and Zita, if it doesn't work, please let me know, because I cannot see anything once I do that. Okay, can you see it?
All perfect, Ruslan.
Okay, great. All right, so today is March 8th also, so International Women's Day, so big congratulations to all women. So I will start with just a couple of points about complex systems, and then I will go into the main part of presentation. So there is a strange field of science called complexity theory, which nobody really can define, even people who work in that field. But it has many fundamental perspectives that are universally applicable. And it's always good to have at least general sort of knowledge of the concepts that are developed in that field, which mostly comes from physics. So in a nutshell, complex systems are made up of multiple, diverse, interconnected components. In the terminology of complex systems, sometimes they called agents, and that interact with each other according to some rules. So that's a very basic description of a complex system. And if any of these conditions are not met, it's not a complex system. If you have, let's say, furniture in the room, is not a complex system because the components of the furniture, they're not interacting with each other. What is really counterintuitive is that the interactions between the agents in the system typically follow a small number of simple rules. So that is very goes against intuition when we think about complex systems, that there has to be something very, very complicated by the way it works. But it turns out that it's usually the smallest possible number of simplest possible rules that govern the behavior of the system. And the consequences of these interactions can be very unpredictable and very, very complex, as illustrated by these couple of examples. So this is a school of fish, and this is the flock of birds. And the school of fish has this beautiful, wonderful sort of shape that continuously moving in circular fashion. The same with this flock of birds. They have these murmurations when the shape changes dramatically. All of you, I'm sure, saw the videos of that. They're really breathtaking. So this is, on the surface, it looks like it's a very complex system. Imagine the amount of information necessary to describe these structures, not to mention dynamics of the systems. But it turns out that they can be understood and described from this perspective I just mentioned, that you have to define what are the agents and what are the rules of interactions. And if you take, for example, flock of birds, so agents would be individual birds. And no matter how you study individual bird, you can know everything about it. You cannot predict these types of behavior. But what you can do is define the rules of interactions between individual birds. And it turns out there are only two rules that they follow. One is that stay not too far, not too long from your neighbor. And two, fly in the direction of average vector of your neighbors. And that's it, two rules. These are very simple rules. There are very few of them, but execution of these rules by all the birds in the flock results in these kind of complex structures. The same is true for fish, of course. So that's a very, actually very profound and important point, that you can have very simple types of interactions between agents to generate infinitely complex structures. And again, the information about the final structure did not exist before these birds started interacting with each other. So that brings us to this point that there are three types of causalities that we can distinguish. The linear causality is what we always kind of seek in nature, and it doesn't exist, actually. This is what the reviewers ask you to do, show the mechanism that A causes B, that it never works this way. More complex and more realistic, the various types of nonlinear causalities. But what I want to emphasize here is the third type, which is the most important here. It's a causality that is, so here we have A causes B, here A causes B and B causes A, when you have this some kind of a feedback. Here, A and B interact. They're not causing each other. They interact with each other, and the consequence of interaction is C. C is something that emerges as a result of this interaction. So this is an emergent causality. And emergent causality is a common theme in complex systems. And thinking in terms of emergent causalities is what, by and large, is missing in biology. It's much more common in physics. Physicists embraced it and chemists embraced it long ago. In biology, we're still trying to fit everything into this kind of a box of linear causalities. Linear causality is fine for the purpose it may serve, with doing reductionist science. There's perfectly fine. There's nothing wrong with it. But we need to understand that the full picture requires thinking in terms of emergent causalities for any kind of a complex system we want to study. So related to this is this point of difference between blueprints and self-organization. Complex systems often exhibit self-organization, resulting in some emergent properties. But there is a common misconception that biologists sometimes fall into when they think about how information is, the nature of biological information. So first, let me explain what I mean here by differences between blueprint and self-organization. So this is some building. This is actually a Sterling Library at Yale University, but it could be any building. To build a building, you need a blueprint, and blueprint contains all the information about the final structure. So it will have every detail of how the building is to be built. And without it, you cannot do it. So information here pre-exists in the form of a blueprint. This is a termite mold. This can be very complex structures. They actually can be enormous in size. They're highly efficient. It's not just a pile of dirt. They have very efficient architecture, which is optimized for this purposes of termite family. It takes sometimes years to build one. Termites only live, I don't know, maybe a week or two. So individual termites have no idea what they're doing when they build this. And there is no blueprint. There is no information that describes what the final structure is. And all that termites do, they follow a few simple rules of how they interact with each other. So they follow very simple algorithms: pick up a piece of dirt, look around, in your vicinity, put it somewhere where there is another piece of dirt, and repeat. And then do that in a way that you always put that piece of dirt in the pile that is the biggest in your vicinity. They don't know the global information. They know only local information, what's in their vicinity. But if that algorithm repeated again and again, as a result, there would be a mold. Of course, there are additional steps there that involved in how the building proceeds. But that's what they do. And by doing so, there is this structure. And this is a very common sort of scenario in biological systems where agents, be it termites, other animals, or it could be cells, interact with each other according to some rules, and what emerges as a result it was not previously described in any form. So similarly, genome people say sometimes it's blueprint. It's not a blueprint. Genome doesn't contain information about the final structure of the organism. It contains instructions how to make it. And the difference is that, of course, imagine how much information is needed to describe a human being, let's say all the neuronal connections. It's an astronomical amount of information. That information does not pre-exist. It's not encoded in the genome. What's encoded is the basically instructions how to make it. What's encoded is the rules of interactions between agents, between cells in this case. So my final point on this part is, I feel is very, very important. When the typical picture is that you have a system, let's say the system always has defined by whatever agents make it up, let's say atoms. And atoms can interact with each other according to some rules. These are rules of chemistry. As a result of that, you get molecules. Now, molecules can also interact with each other according to some rules that include chemistry, physical chemistry, colloidal chemistry. There's various types of rules that different disciplines study. They can polymerize, for example, and they make macromolecules, and then they can interact with various types of rules of complementarity to make macromolecular complexes. That, so far, everything looks fine. We can say, well, this is one level, this is another level, another level, another level. At each level, the agents are different. Here's atom, here's molecules, here's macromolecules, here's these complexes. But then something happens and boom, and we have cells. But we don't actually know whether between macromolecular complexes and cells, whether there are additional levels of organization. And one would argue that, of course, there must be, but we don't know what they are, and we don't even know how many there are that we are missing. And whenever you have this kind of a gap, it always gives this sense of mystery, something mysterious that we cannot explain. It's not that we don't have enough data. No, it's something beyond the data. So it's two types of unknown in science. One is unknown because we just didn't get enough data yet, but there is nothing mysterious about it. And the second type of unknown is of this type, when there is a missing level of organization, missing level of emergence. We don't know what the agents are at this level. And from there, boom, we have cells. Now, okay, we now, again, in our comfort zone, we know cells are agents. They interact with each other, they communicate, and then somehow we get tissues. But here again, we have a mystery. How do you go from cells to tissues? We don't understand that. And again, that is a sign that there is another level of integration, level of organization that's missing. When cells interact with each other, it doesn't result in tissues. It results in something else here for which we don't even have a name. And these agents, when interact with each other, make tissues and so on. And then there is another level perhaps missing here. Then you get organs, organisms, and populations and so on. So these missing levels of organization is what gives this sense of mystery. And in the past, it was filled in with the idea of vitalism, but now we need to explain it in scientific terms. And I would argue whenever we have that sense of mystery, it's a wonderful, of course, feeling, but it always, I think, implies that there is a missing level of organization. And of course, by definition of emergent properties, at each level, you cannot describe the system at a given level by knowing all the details about previous level. You can know everything about atoms, hydrogen and oxygen. You cannot predict properties of water molecule. And that's true in all cases. So that means also that when we study, let's say, tissues, just knowing everything about cells is not enough to understand tissues. We need to know the rules of interactions between agents at the previous level, and this way, just like we know rules of chemistry to understand molecules. So that's the second point. So now I will start talking about more biological examples, and specifically about some design features with an eye on mostly on immune and nervous system and looking at parallels. I should just say that at this point that one of my pet peeves is that I feel like the biological disciplines like immunology and neurobiology in particular, we don't understand them from first principles. We understand biochemistry from first principles, genetics, evolutionary biology, but developmental biology, neuroscience, immunology, we don't understand from first principles. And that is always going to be a limitation in the advancement of the field. And what I mean by first principles is that if you try to explain, I mean, this is something that's often encountered if you teach introductory courses, right? When the types of questions that students would have and ask, they relate to questions of first principles. And this would be the most basic foundational questions about the system. And as soon as we start addressing them from that perspective, things become much better, much more intuitive and better connected between different fields. So if we think about, so this is the sort of motivation, the way I will talk about this part, which is really thinking about it from first principles. So we can think of a system, could be biological system, could be cell, could be organism, and it can perceive some external stimuli to produce some response, and it can also detect system state. So it would be internal stimuli that can also generate some response or modify response to external stimuli. And the way that systems are designed generally is that, so we have several types of input characteristics. The source could be either external or internal. The internal would be system state. They have salience, and that means that the capacity of the sensors that can detect this stimuli, what is detectable, either outside or inside. They have valence, the meaning of these inputs, and that's related to system's goal, right? So if the smell of a rotten banana would have negative valence perhaps to us, but positive valence to some fly. So valence related to the system's goal. They have dynamic properties like amplitude, frequency, resistance, and they could be either cues or signals. So everything is simple so far. So the stimuli can have these characteristics. What system responds to detect relevant stimuli needs to have sensors. Sensors of capacity determines what is detectable, like their salience, and then there is some meaning assigned to it. And this, what I mean by cues versus signals is that it's an important distinction between information carriers. Both signals and cues transfer information, but the difference between them is that signals are generated in order to transmit specific information. And the cues, on the other hand, are not generated with any purpose. Certainly not with the purpose of transmitting any specific information, but rather they are proxies for something that cannot be detected directly. And they are associated with relevant information in the environment, information that have some salience and detectable by the available sensors. So the difference between signals and cues, let's say you hear let's say sound of the steps or sound of the car that parked at the garage. Then you know that there is or just the sound of the car driving, the motor engine. That tells you, well, there is a car approaching, right? But the engine doesn't make sound to tell you that the car is approaching. It's just the consequence of the working engine. On the other hand, if it honks, that signal is made on purpose to let you know there is a car, right? Let's say the sound of in the forest of a crouching tiger, right? That would be a cue to some antelope that there is a predator and it will run away. Of course, tiger doesn't want to make that sound, It's just a consequence of it walking in the forest. But for the antelope or I don't know, a rabbit or something, that is a cue that there is a tiger, and it will use it. It gives that information that there is a predator, and it will run away. And this is in contrast to tiger making, let's say, mating sound to attract mates. That sound is made on purpose to transmit specific information. So signals often, if not always, evolve from cues. So here's an example with ATP. So ATP can be released from dead cells when they die by necrosis. And then macrophages and other cell types, mast cells, can detect that ATP and say, oh, there is a dead cell. We need to induce some inflammatory response. So here, ATP is not released with the purpose of transmitting an information. It's just released because the cell died and membrane integrity is lost. On the other hand, ATP can be used by certain sensory cells, like taste sensory epithelia, and they release it as a neurotransmitter. And they release it in a controlled fashion to activate neurons downstream to transmit information about taste, for example. So here, ATP is used as a signal instead of a cue. And if you think about various neurotransmitters, many of them you can recognize what they derive from, and whatever they derive from, for example, by decarboxylation reaction, like the way you get histamine from histidine and serotonin from tryptophan and so forth. In all cases, you can see how the original message was something that presumably was just a cue, and then with some modifications became a signal. So similarly, that's what I mean here, that what's detected by sensors could be cues or signals. And depending on that, there would be different characteristics, like when the detection of odorants, for example, odorants are cues, whereas pheromones might be signals if they produce on purpose. That would be the differences. And so the sensors that detect the stimuli, and again, I'm talking here very generally, the system could be a cell, it could be an organism. But I mostly will have organism in mind because I want to transition to immune and nervous system parallels. So sensor characteristics could be, they could be tonic versus phasic, so they have or don't have adaptation. They can be optimized to either minimize type 1 or type 2 error. And what that means is that there is a trade-off between detection of between false positive and false negative rates. So what kind of error the system makes. And because of the trade-off, it's basically trade-off between sensitivity and specificity. And so the high sensitivity means there is a low false negative rate, and high specificity means there is a low false negative rate. And which of this is optimized depends on the valency of the stimulus. If the stimulus has negative valency, then what's optimized is sensitivity over specificity. And the reason for that is because it's better to be wrong than sorry, right? That's the idea. And for positive valency, it's the opposite. So there is this important distinction between them. And then there is a tunable gain of the sensors. And the gain of the sensor means what is input-output function. For this much input, how much output is generated. And that is defined often by the state of the system or by the memory. For example, if you're hungry, the smell or view of food you will be more sensitive to the smell of, for example, high-calorie food if you're very hungry. If you're full, so that system state changes, now you will be less sensitive to it, or you stop reacting to it altogether. And again, sensors can be defined by whether they're sensing cues versus signals. And you can imagine sensors that detect signals are far more optimized for signal detection because you can evolutionarily tune affinities and so on. Whereas in the case of the cues, we are at the mercy of what are the available cues. They cannot be controlled by the system. So The second point is that the way that the system is typically designed with regards to the information transfer is that the parts of the systems are dedicated to detect the stimuli, internal or external stimuli. These are sensors. And the other part of the system is dedicated to make changes to the system that correspond to the meaning of the stimulus. So this would be some kind of an effector part of the system, which will induce some kind of response. So this is one simplest possible design where you have sensors in effector and they connected by some signal. And for example, all homeostatic circuits are based on this design. You detect something. If you don't like the value, you activate effector to change that value in the right direction. The second design is where you have the same essential components, sensors and effectors, but in between you have an additional component that collects information from multiple sensors and can control multiple effectors. And the terminology here now becomes arbitrary. I put the term controller here because that's what's used in engineering. And in biology, people use different words for this. Details don't matter, the design matters. So design is always looks like this. So this is the second type of design. And so the immune system in nervous system use both designs. So sensor effector, simplest design. This is how innate immune system works. Macrophage is a sensor to some, let's say, stimulus being pathogen, make some signal to activate effectors, in this case, neutrophils that will kill pathogen, and it's done. So that's one design where there is direct connection between sensor and effector. And the second design when there is this controller in between, and the controller is always a lymphocyte of T-type lineage, either ILC or T cell. So controller in the immune system in these circuits is always T-type of lymphocyte. So again, you detect sensor by some APC, it makes some cytokine, activates this lymphocyte, they make their own cytokine that activates effectors. So that's a design of the adaptive immune system. And in a nutshell, I would say if I was asked to describe the immune system in 2 slides, those would be those 2 slides, that this is how it's designed. Everything else is variation on that theme, including this very sophisticated function of the adaptive lymphocytes that have antigen specificity and so on. All that is superimposed on this simple principle. Now, in the nervous system, of course, there's a very similar designs. The direct sensor to effector connection, so sensors would be sensory neurons and effectors motor neurons. This type of design is common in more simpler nervous systems, and more common design would be to have a controller in between sensory and motor neurons. And the controller part in the nervous system would be various interneurons. And typically more than one, because the point of the controller is perform some kind of computation to integrate information from various sensors and to decide which effectors to activate or inhibit. So it usually would be some neuronal circuit that is made up of interneurons that get inputs from sensory neurons and control output from motor neurons. And basically, the nervous system, as we know it in complex animals like humans, most of it is elaboration of this part. The entire CNS is basically elaboration on the idea that you have something between sensor and effector. There are sensory inputs that are relatively simple compared to CNS. There are motor neurons, relatively simple compared to CNS, but majority of elaboration happens at this level. In contrast to the immune system, where we stop with lymphocytes, in the nervous system, that's just the beginning. And one can imagine these levels of organization. So we have the simplest one, sensor to effector. We have a slightly more elaborate where you have a controller in between, and that collects information from multiple sensors, performs some computation, and controls effectors. So it's either T lymphocyte in the immune system, or it's some interneuronal circuit in the nervous system. But in the nervous system, it doesn't stop here. There is a next level of elaboration where you have something between controllers. let's call it integrator. The term is not important. It's intuitively obvious that the difference is between them. So now the integrator would be collecting information from controllers. Each controller collects information from multiple sensors again and controls multiple effectors. And the point of the integrator is the next level of organization. So now we have integrator collecting information from multiple controllers, right? So now, as you recall the beginning of the talk, when I was talking about levels of organization defined by the agents involved. So at this level, agents are controllers, and at this level, it's integrators. And in the nervous system, this process goes on for a number of steps. We don't know how many steps involved, then connection between integrators and so forth. And at each level, we have some emergent properties that are generated. There are new types of properties, including computation, memory, and so forth. And the way it would look like schematically, so let's say these are all these different levels. We don't know what they correspond to in each case. So at some point, for example, you go from interneuronal circuits, then they organize in some ganglia or brain nuclei, right? So this would be corresponding perhaps to them. And then we have these additional properties that need to arise, that these next higher levels of organization now they're dealing with much greater complexity of inputs, and they need to filter the inputs. And filtering of inputs maybe perhaps would be some function, primordial function that later became elaborated into thalamus that controls what inputs go in and which don't. And then you also need some you also need some kind of a memory device, and then you need some additional components at this level that allow to the system to function properly, recording the internal states, for example, and so forth, valency, and so on. And that's sort of the type of elaboration that I wish there was a textbook of neuroscience that described it this way. I'm not aware of one, but I feel that would be a description for first principles, regardless of any kind of oversimplifications and little mistakes I made along the way here. But the idea is to understand it from that point of view, and to understand why do we need both amygdala and hippocampus for memory? Why do we need, well, in this case, we can say it's the memory with valence versus contextual memory. Why do we need thalamus? Why do we need parabranchial nucleus? What's their essence? What's the point of having these types of brain regions? And that can all be ultimately understood from these kind of first principles, where we progressively increase levels of, or go from one level of organization to the next to understand its meaning and its essence. So the input characteristics to the system, again, could be external or internal. The external ones are detected by special senses, taste, olfaction, auditory, and so forth. They are typically conscious because they need to be connected to behavior. On the other hand, internal ones are detected by visceral sensors. And here we, of course, unlike special senses, which we know all of them, for visceral sensors, we don't know all of them, and we don't know what they're detecting in many cases. We know some of them, what they detected, but we don't know how many and what is detectable. And that is the whole point of body-brain interaction, to understand that question. They are only conscious if behavioral response is required, otherwise they're not, but they can indirectly affect behavior through emotional state and by tuning the gain of external sensors. So that's the connection between them. And now I would want to turn to parallels in learning between immune and nervous system. So with the nervous system, of course, the simple paradigm is associative learning or Pavlovian conditioning. You have what is key here, we distinguish between two types of inputs. There's unconditional input. So this is now we're talking about the types of stimuli that can go into the system. There is an unconditioned stimulus for which there is a hardwired response. and there is conditioned stimulus that is neutral. It doesn't elicit any response. We can say that this stimulus has a meaning. It means there is food. This stimulus has no meaning. We can detect it because it's a sound or whatever by other special senses, but we don't know what it means. When they're associated, then the meaning of this stimulus is assigned to this stimulus. And if this stimulus is more convenient, better detectable, and so forth, then it becomes a stimulus of choice to report on the presence of the food, for example, in this case. So that would be conditioned response. In the immune system, there's exact same principle that operates in connecting conditioned and unconditioned stimuli. So what is unconditioned stimulus in the immune system? These would be the structures detected by the innate immune system through various receptors that detect things like LPS, peptidoglycans, and so forth. So that induces hardwired pre-programmed response. The equivalent of conditioned stimulus would be an antigen. It has no meaning by itself, and by itself it elicits no response. However, when they are combined together, then the meaning of these microbial structures, meaning in this case would be the microbial origin, is assigned to this neutral stimulus, which is an antigen. So in bacterium, there would be both LPS and some protein antigens, and detection of them together, associations between them, results in immune response that now can be elicited against antigen alone. So this would be now conditioned response, very much like Pavlovian conditioning. Mechanistically, of course, they're different, but the idea is the same. And what I would really want to draw attention to is that what happens in both cases is that the meaning of unconditioned stimulus, which is determined on evolutionary time scale. On evolutionary time scale, we know this, how to interpret the smell of food or what LPS means. It means infection. But then this meaning is assigned to something that has no meaning, that's neutral, has zero, has no valency. And as long as it has some kind of a detectable structure, the meaning of unconditioned stimulus is assigned to it, resulting in this associative learning. So the way it would work, for example, for immune system, you detect antigen and LPS or other molecule, some dendritic cells become activated, and now these dendritic cells tell T cell that this antigen here, now it has a meaning, and it turned red. So it was gray here, now it turned red because it was recognized in the context of molecule that is unmistakably bacterial. So now it says this thing here is bacterial. T cell has random T cell receptors, doesn't know what they can recognize, but when it detects this antigen, this dendritic cell tells it this antigen came from bacteria. Now the T cell became activated. Now it knows what its receptor is specific for. It's specific to bacterial antigen. And then it goes on and then encounters B cell that has a receptor that is random. It detects this antigen. It's gray because it doesn't know where it comes from and what it means. But because it will present this peptides from this antigen to T cell will tell us, this one comes from this red antigen that comes from bacteria. And now B cell makes antibodies to this antigen. So in this way, you can see in all cases, we have association between neutral stimulus and meaningful stimulus. And the semantic assignment from meaningful to neutral one then continues in several steps from dendritic cell to T cell, from T cell to B cell, and so forth. And that's the way that learning happens in the immune system. Now, in the nervous system, again, I would like to see a simple schematic that I'm just that would allow to make a simple similar point. But I just made one. It's maybe it doesn't correspond to anything, but it's something that like the circuit in aplesia conditioned response. So we have some unconditioned stimulus, like for example, shock, detected by some pain receptor. So this sensory neuron then connects to motor neuron to tell to induce muscle contraction and withdrawal. Right, so that would make sense. So this is a hardwired response. There's something bad detected here. Appropriate response is some kind of a movement. And so this sensor detects something that has meaning, in this case, pain, for example, or it could be some chemical. On the other hand, there is another neuron, another sensory neuron that detects conditioned stimulus that has no meaning, some, let's say, touch, or some molecule that has smell, but we don't know what that smell means. And then this conditioned stimulus, if encountered at the same time as unconditioned, you have this some kind of facilitation or whatever. There's different ways that this could work, but I just made it very simple here. You can have, let's say, facilitation and resulting in conditioned response to this stimulus after this association happened. And in the case of the immune system, recall that the receptors for unconditioned stimuli are germline encoded receptors of the innate immune system. Receptors for conditioned stimuli being antigens, would be antigen receptors of T and B cells that are randomly generated and have no pre-existing, sort of not predetermined meaning. And their association then results in meaning. So there we can say, well, there are pattern recognition receptors here and there are antigen receptors here. And in the case of the nervous system, similarly, the question would be, what is the difference between this sensory neuron and this sensory neuron, right? Well, of course, they will have different modalities, perhaps, but what is the difference between them in terms of the design of the circuits? And because one can say that the sensory inputs that detect unconditioned stimulus, hardwired stimuli, and the ones that detect neutral stimuli, which need to be assigned some meaning, their design would have to be different. And what are the general rules of these differences in design between the two types of sensory neuron would be a question that I've been struggling to find answer anywhere in literature, but perhaps it's known and I'm just not aware of it. Now, one of the last points I want to make here is that the way that the both immune and nervous systems operate is that they in both cases, there is this related process of generating something that didn't exist before. For example, antigen receptors are generated. They're not pre-existing, not genetically predetermined. Similarly, all synaptic connections in the brain, neuronal circuits, they are not predetermined. They are generated de novo, and that information doesn't exist in the genome because it's too much information. It cannot be stored in the genome. And the way it works is that there is always some general principle of how information is generated is that you have randomizable precursors, for example, fragments of antigen receptors that then can be combined together. So it's a randomizable precursors, V regions of T and B cell receptors. Or these could be different synaptic connections that are generated at random. So then you have a random set, and then you have selection, antigen receptor selection in the immune system, or synaptic connection selection in the nervous system. Both of them operate according to their own criteria of selection, like matching neurotransmitter and receptor and activity of the target, or matching TCR, MHC, specificity, and so on. But as a result of selection, you have something that you have information, enormous amount of information that didn't exist before. Millions of TCR specificities, billions or trillions of synaptic connections that were not pre-specified. But once they're generated, that information is there, and that didn't exist. And the way it was generated is by making randomizable set, taking some randomizable precursors, making a random set, and then followed by selection. The size of this random set determines how much information you can generate. The criteria for selection determine the meaning of this new information or their semantic content. So this would be can be illustrated like this. You have random set, and you have a set with assigned valency or assigned meaning. You take one element from here, one from here, and you do some kind of a association between them, either temporal or physical or other type of association, depending on the system. And then you assign meaning of this stimulus to this neutral stimulus, and now it becomes red because that's the meaning here, and now it becomes part of this set. Now, since it became part of this set, now this stimulus can now be used to associate with something else. And now you assign meaning from this guy who was previously neutral to some new neutral and assign the meaning to it. In the immune system, this process goes only a couple of steps in the phenomenon called epitope spreading. In the nervous system, it's almost unlimited. These would be different levels of abstraction, how many steps you go from assigning meaning from newly assigned signal to neutral signal and continuing in this way. This would correspond to different levels of abstraction, and we don't know where the limit is. And so I will just finish here with summarizing this part, that universal steps in generation of new information is making new set, selecting from it, and selected elements of a random set are assigned meaning by one of the possible mechanisms through association or reinforcement, and the semantics of the new information is defined by the selection criteria. So I will. I think I will stop. No, I'll just this last slide. Stop. So this is some comparisons between the properties of stimuli for the nervous and immune system, and I think I just want to make this correspondences. What we call salience in the nervous system is antigenicity in the immune system. The aliens here would be immunogenicity. We have both external and internal inputs in the nervous system. We only know of external inputs for the immune system, but there's growing evidence that there are also internal inputs that are relevant. Both use associative learning, both use this generative information that's not pre-encoded. Circuit hierarchy here is, we don't know what the limit is. Whereas in the case of the immune system, it's just sensor, controller, and effector, controlling being lymphocyte. And the computation in the case of the nervous system can be both central and distributed, whereas in the case of the immune system is distributed only. And that's my last slide, is to say that the two types of computation, centralized and decentralized, have been defined by John von Neumann. Centralized is what you use, for example, by computers with CPU and based on local and global information. Decentralized is exemplified by cellular automata, is based on local information only. And the nervous system has clear centralized control by the CNS. However, the enteric nervous system appears to be based on decentralized control. This is just my hypothesis. This is not a statement. It's just a question that it's based on decentralized control, but may be subjugated to central control by the CNS. And similarly, immune system has decentralized control only, but may be subjugated to central control as well by CNS, as Asia's recent work beautifully demonstrated by the impact of insular cortex. So I'll stop here, and thank you very much again for your attention and for having me here.
Thank you very much for this great and very inspiring talk. It's always really challenging, I think, right, to organize this never-ending data stream, which we all feel subjected to in biology, and trying to see, right, what are analogies, what are general principles which might be underlying all these complex systems. So thank you very much for having done this really challenging and I think it's important exercise with us. So what we had thought we would do is we would have a small, we would change a little bit the pattern where we normally have the moderator pick up questions for the audience, and we will have this time people who are from the organizing system, from the organizing committee here, to discuss a little bit with you and have some questions. Obviously, we will try to also integrate some questions from the audience. And well, I would like to start maybe by asking the first question, Ruslan, and that goes back to your first, to your beginning of your talk about this mystical step, right? This question mark, right? These unknown organizational principles. And it also goes back to a question asked by Tom, I think, right? Which is, I mean, how do we how do we identify a level of organization as being a level of organization, right? Why do we say cells are a level of organization and organelles maybe might not be taking that level of organization? And I mean, my intuition is that many of the things you had on that slide can be anatomically, therefore visually identified. But do you think that we need to go beyond that and have other ways to identify these levels of organization? So how do we go about filling in these question marks?
Yeah, no, that's I think is one of the really key problems in being able to define the levels of organization. The way they're defined by the relevant agents of the system. Like in simple examples would be atoms is agents on one level, molecules on another level, and we know how you get from atoms to molecules. There is no standard procedure to tell that there is a missing level of organization, and there is no level to tell if something is an agent of the other system. But the way we generally can tell if that's the case is by identifying an entity that has an emergent property from interactions of other entities that generate it. And that's not always clear cut. It's not always straightforward. In the case of molecules, atoms, cells, individual organisms, it's obvious that those are the entities, those are the agents. But and that's why they've been defined so easily and so early. But in many cases, especially subcellular levels of organization, just one level before cell or two levels before cell, there we don't even know where to start. And I would say that in my mind, the only way to detect it is by having that sense of mystery. When some phenomenon, we know that we can observe it, we can see it, and we look at it in the video and microscope and say, wow, how the hell does this happen, right? How it knows to go here and to do this. When we have that sense, that means that there is a level of organization missing. But I'm not, to answer your question, I don't think there is a standard method to do it other than having that moment of wow, how does that work? Whenever it elicits some kind of a almost religious sort of awe that something is happening there.
So it's kind of using our brain as a pattern detector, right? As a
yeah. In this case, yeah.
I mean, maybe actually that's an interesting way of thinking about one thing that brain needs to do is to detect levels of organization and simplify the world in doing that, right? I mean, maybe that's why we have this sense of like, wow, something interesting is going on there, right? Because ultimately we need to decompose the world into understanding how it works, right? And maybe we have evolved to be very good at that. Maybe that's what the brain also does, right?
Yeah.
Jerica, what?
Yeah, can I chime in? Something you said, Professor Ruslan, Medzhitov, you mentioned it's hard to know like what is an agent of a system, and it made me sort of think of like the crosstalk between our just like broadly neuroimmunity and how we're reusing different systems like cytokines or receptors in both the nervous system on neurons and in immune cells. And is this just a fact of evolution? We've reused different molecules or proteins in different systems in different ways, or is this actually sort of a smart move of our biology to use these sensors in both systems? Or not sensors, I was thinking.
Yeah, it's a good question. So one way to think of it is why the two systems have to interact, right? Is it just trivial reasons like everything interacts? There is always some kind of a connection. They're part of the same organism, or whether there is a deeper reason. And I of course would think that there is a deeper reason for the interaction. And I will give just a couple of examples. why I think so. One has to do with one would be from the perspective of sensory modalities. So imagine nervous system has sensors, let's say for external environment, for chemicals and for sound and vision, so it can detect that. It can detect multiple molecules. There are, like maybe hundreds of odorants can be detected. Similar number of different tastes by combining different taste inputs. But it's not unlimited what can be detected by the nervous system. On the other hand, the immune system, as long as the input has an antigenic structure, meaning it has some, let's say, protein detectable by T cell, then what can be detected is unlimited, right? On the other hand, so in this way, I think of these two systems as complementing each other in terms of sensory modalities. So you need to detect 100% of relevant inputs. One system can only detect, let's say, 30%. The other has to be complemented by the other system, by the immune system, let's say. The other example would be that when we think about this, what I mentioned about the complexity of these levels of organization, when you have sensor and effector, sensor control effector, and so on and so forth. If we think about, like, at some point there is this requirement for memory and like various computations that nervous system can do, but an immune system cannot do. And then perhaps the immune system takes advantage of that ability of the nervous system when it's necessary. And the reason this has never been sort of brought up as a question in the immune system, whether there is a necessity for additional type of computation, is probably because it never came to, we immunologists were always satisfied with how we define immune memory, right? So immune memory basically defined in a very simple way that response to first exposure different from response to second exposure. But the question one can ask is, what is stored in immune memory? What kind of information is stored in immune memory? We know, well, what is stored, of course, antigen structure, because that's what the point of clonal expansion is, detect antigen structure. So that information is stored in the immune memory. What else? The type of pathogen also stored, because there would be different types of T cells. Location of the antigen would also be stored, because to some extent, it will say it's a gut versus lung or skin. But beyond that, we don't know what else is stored, what kind of information. And what I think is that immunologists became sort of content with the notion, well, you have this immune memory, it's wonderful, it works. But we actually don't know whether it stops there or if there are additional type of information that need to be stored. So let's say there is a viral infection, you induce some response, and that response was unsuccessful. Would that information be stored? The fact that it was unsuccessful, or the fact that if you induced response A, it was much better than response B. You'd think that, well, for a smart system, you probably would want to remember that, right? But we actually don't know if immune system does store that information. And again, ASA's recent study suggests to me maybe some of that is delegated to the nervous system, because immune system doesn't have the capacity for that beyond like a few things I mentioned, and maybe it delegates that to the nervous system. So these are two examples which, to me, imply that there is a much more fundamental reason for interaction between the two systems, not just one modulating the response of the other, but rather there are more sort of fundamental reasons from the system's perspective. You're muted.
Sorry, it didn't click. Yeah, thank you. That's definitely interesting. And another thing that makes me think further as like a mystery is like, why are certain molecules like complement or like cytokines used in the nervous system for a different function that's like completely separate from like an immune challenge or activation? So thinking very much to like developmental synapse pruning that uses complement, for example, but other molecules as well. Just something that always sparks my thinking.
I think my my thinking about that is the concept of exaptation. So the concept of exaptation, which was proposed by Good in 70s, I think, is an evolutionary concept that some trait evolves for one purpose, but then can acquire additional function, and that function may become dominant and optimized, and then you don't know what the initial function was. So exaptation is basically exploring all the possibilities. And so the famous example being feather. Feathers didn't evolve for flight, they evolved for thermoregulation, but because they're also good for flight, that function is optimized, and now they're actually better for, well, some feathers are only good for flight. So that's an exaptation. So take complement. The complement evolved, let's say, to remove, let's say, bacteria. But the way it works, it just tags something that you want to get phagocytosed. So it could be bacteria, it could be dead cell, it could be unwanted synapse, it could be unwanted anything that you just need to tag to remove. So that would be an example of exaptation. And the immune system, I think, and nervous system, they both have a lot of examples of exaptation, that something evolved for one purpose, but it's actually such a good, it's like a wheel, right? The wheel was not invented for airplanes, right? They didn't know about airplanes when the wheel were invented, and not for cars either. But now it became so obvious in the case of cars. We think, well, wheels are for cars, but initially there was nothing to do with cars. Similarly, some of the things we think, oh, complement is important for bacteria, but maybe initially it wasn't for bacteria at all. The same with phagocytosis, with cytotoxicity, and many other functions that we, as well as function of many cytokines. What did TNF evolve for? Well, we know it mostly in the context of infection, but this may or may not have been the initial purpose.
Sure.
Okay, now we switch to Zita. I think you wanted to introduce a question for the audience.
Yeah, Ruslan. So we have a question from Miguel Aliaga, and so I will just read it to you. Great conceptual talk on the difference between signal and cue. To me, it sounds a bit designy. It makes it necessary to invoke purpose, and perhaps the difference is less clear-cut when considering, for example, instructive roles of metabolites. Could one not see it instead from the receiver's perspective? What relays a message and is ultimately informative, instructive in terms of responses is what was limiting in the first place, chemically or otherwise. What do you think?
Well, I think the difference between cues and signals, as defined by whether they are produced on purpose, that definition works in most cases. Where it breaks down is when we have cases of the same molecule having both roles, like I mentioned with the example of ATP. And then we need to define whether, and in the case of ATP, we know that, but in other cases, we may not know enough biology to tell the difference. For example, lactate made by proliferating like cells that have Warburg glycolysis. And then lactate can be interpreted by other cells. Is it made with the purpose of informing other cells, or is it made as a byproduct of aerobic glycolysis? We would probably, our first reaction would be say, well, it's a byproduct of aerobic glycolysis, therefore it's a cue, not a signal. But what happens often with these cues is that if they're really good, if they are really good representation of some process, in this case, glycolytic metabolism or cell proliferation or tumor cell, then they can evolve into a signal. And that usually requires, if we think about all these biological signals that are based on small molecules, usually requires that generation is controlled, for example, release by exocytosis like ATP, or that it undergoes some modification that takes it out from its normal context. So for example, it's often would be decarboxylation of amino acid to make a neurotransmitter, or acetylation to modify it so that it's now not part of the normal metabolic flux. So it takes it out of the normal function And this modification is what's regulated. Either modification itself or release is regulated, and this way you get a signal out of a queue. But I agree that there are gray areas where, but I would say that these gray areas are basically limitations of our current knowledge, where we cannot tell. Like currently we think lactate is a queue, not a signal. Short chain fatty acids are cues from microbiome, they're not a signal, but perhaps some bacteria actually decided, okay, I'll make it on purpose because that will make my life easier if I calm down the inflammation in the gut. So that would be the kind of area where we would be limited by how much we understand the system.
Fantastic. Ruslan, maybe I can ask you just one short question, and then I'll give my word to Vajia. So I was wondering if by just studying these principles and these levels across evolution, what can we learn, right? So can we learn about what are the basic principles? Can we learn more about which could be other levels, or by just looking at what are the components which are common between a bacteria and a cell in a multicellular organism, or by just looking at the steps in evolution where you go from one to multiple cells. So what do you think, at which level can we learn by looking at across evolution?
Well, I would say that I really like this quote from a book about the system science. And the quote is that every complex system is derived from a simpler system that worked. So in evolution, that's true for everything, right? Everything is a modification of something that already existed and it worked. Otherwise, it wouldn't have existed, wouldn't have been stable. So no matter what we're studying, and no matter what level, no matter how complex it looks, including things like the brain, there was a simpler version that actually worked for what it needed to do. And the question is always, what is that simpler version and what's the simpler version before that? That's one way to go, like top down, to think about it in this way. Another way is bottom up, is to say, well, in the beginning, you just have some sensory inputs and some motor response. That's what nervous system is, the simplest nervous system. That's all it does. And it's actually, in the beginning, was decentralized design. There was no CNS. It's just a neural net, right? And then from there, you go and start asking, what were the forces to select for modification of that design, that now you have some integration centers, or you have some simple computational circuits by interneurons connecting sensors and motor neurons. And going from the bottom, and like just the way I tried to schematically illustrate with a cartoon, you make more and more these intermediate levels. And then in each step, you ask what additional functionality it brings, right? So you can imagine on the plane, there are sensors for altitude, for speed, for temperature, for whatever, the position of different sort of regulators of the direction on the wings. All the sensors collect information and comes to the computer on the plane, and then computer then gives signals back to them to modify direction, altitude, and so forth, right? So that would be autopilot. So you have sensors, they have central computer, they have the motor outputs. But then on top of it, you have a pilot, right? So pilot will look at it and say, well, the autopilot says continue flying at this altitude in this direction. And then autopilot has some other information that says, well, actually, we need to turn because there's turbulence coming. And it overrides the autopilot and changes altitude, let's say. But then autopilot sits above the controller. Controller would be the autopilot. The pilot is above it, right? So this what I try to illustrate, something sitting on top of it. Why do you need that on top of it? Because of what I just mentioned, because of it has information that autopilot doesn't have. But then the pilot of the plane, let's say, then is connected to central control unit of whatever it's called, that people who regulate traffic, air traffic, and they tell him, actually don't go into that airport at all, just turn to the other airport because there is going to be a hurricane or something. And they override the pilot's information. Pilot overrides autopilot, and autopilot then changes sensor and effector function. And then it goes on and on like this, and then it stops. For airplanes, it stops after 3 steps. But for the nervous system, it doesn't stop at 3 steps, and we don't know how many steps there are. And why at each step you have these additional types of functionalities that result in various brain nuclei, for example, and what was the need for them to emerge in the first place.
Fantastic. Well, now we pass to Asya.
So thank you, and I'm really enjoying this talk, and I have so many questions, the discussion, but and there are many questions actually in the chat. So, but I wonder, in terms of integration, at which level the system does integration? So you can collect information in the level of receptors, right? You can collect information at some level of higher organization, maybe lymph nodes in the case of the immune system. And I wonder, like, what do you think, what kind of levels of integration do you think they're in parallel between the brain, like the nervous system and the immune system? Do you think, like, what is the meaning of having different levels of integration of information that can be detected, I think, by the nerve? Definitely. I even wonder if the immune system might even have higher level ability to integrate different pieces of information.
Yeah, I agree. I also started thinking that it might, that there is something beyond lymphocyte, as in terms of this levels of these circuits, that beyond lymphocyte would be something that we cannot even define. It says, it relates to Carlos' earlier question, how do you know what's missing, right? So for a long time, immunology as a field was very happy that we didn't think there's anything missing, right? And when do you know that something is missing? When there would be some new discovery that doesn't fit with your world view, right? And says, there is something else beyond that, some kind of a decision-making process that orchestrates the whole immune response, and it's more central than just lymphocytes, proliferating or and so forth. I don't, in this case, I don't think that there is a centralized control still, other than within the immune system. By the nervous system, yes, but within the immune system, I don't think there is centralized control. Whatever the next other level organization, if it exists, I think would still be decentralized. Again, meaning that each agent, in this case lymphocyte, operates based on local information only. There is no global view. The global view, I think, is only provided by the nervous system. And I think your discovery actually, I think, is a good example of that, the immune system is limited in its ability to compute certain things. And so it delegates that to the nervous system. And in this way, the two systems, again, can interact in a very deep, fundamental level where the capacities, a limited capacity of one is compensated by capacities of the other. But I agree that there is we never say never, right? In science, it's always possible that there is some other level. What I think is missing actually is that in the case of the immune system, we have not yet embraced these ideas of emergence and complex behaviors that arise from it, right? We have not embraced crowd behavior of agents, of lymphocytes and so on. We only think of them in terms of these cartoons. Dendritic cell activates T cell activates this, linear cartoons. as if that T cell exists by itself. We ignore how it will interact with other T cells and dendritic cell with other dendritic cells. And this type of collective behavior is then lost on us. And therefore, we're happy with cartoons because they're simple and we can understand them. But then we encounter some strange situation, why immune system couldn't get rid of COVID, for example, why it attacks our cells. And we draw this simple sort of cartoons and we hit a wall. And so I think that that's a big limitation currently.
Yeah, absolutely agree.
So, I mean, I was asked to maybe ask you the last question. Obviously, as Arte was saying, there are many other great questions, and we could continue for hours. But unfortunately, we have to bring this soon to an end. But so we've been thinking a lot in this discussion group about like kind of what is required to start thinking more holistically about these interactions within systems, right? What is kind of also what is required beyond just interacting with people thinking about these different systems, more also thinking about education and student training and so on. And so I was wondering, I mean, what your perspective is, right? For example, when I look at your talk, I see a lot of echoes from complexity theory, but also from things like, for example, control theory, right? Engineering principles. So do you think we have to start teaching students not so much mathematics in terms of tools to analyze data, but also mathematics and complexity theory and engineering principles as a way of thinking about systems? Or, I mean, if you could kind of, you know, add something to the training of students to prepare them to think about and discover these levels of organization, what would be your advice?
Yeah, I think there is a couple of problems with the way things are now. One is obvious, that the biological science, well, like other sciences, it's becoming more and more fragmented. And one consequence of this fragmentation is that each subfield then becomes specialized so much that they develop their own language. And that language then is not understood by anyone outside of that area. And then even if they speak about the same concept, they don't know it, that they speak of same concept, if they have similar idea. So biologists, unfortunately, are very, on average, experimental biologists, on average, are very dismissive of generalizations. They like details. Nothing wrong with details, but just knowing more and more details is also not going to get you too far. And what is generally, I think there is a kind of a dismissive attitude towards anything that would be a generalization, anything that would take a little bit of formal language of describing it, a little bit of a right level of abstraction. And of course, this can also be abused to the point that you become a philosopher and then, you know, too abstract and too far from it. It has to be the right level of abstraction, and it has to be the right type of formalism, formal description, the way that physicists and mathematicians use. You don't need to know all the, you know, techniques of some, you know, differential calculus or anything like that to think this way. You just need to acquire this mode of thinking. So in the exact sciences, they move from specific to general. In biology, we move from general to specific. As a major flaw, major trend, we go into more and more specific, and then we lose the big picture as a result. And to get to the big picture, we need to accept this language of formal description of a problem. And unfortunately, the current trends take it even further. Current trends, empowered by new technologies, take this trend even further, where existence of powerful technologies, which are wonderful, of course, it makes it all too easy for somebody coming into science, students and so on, trainees, says, okay, I can use this technique to do this and that, rather than what should I do to answer this question, right? like 90% of papers in top journals are just completely unreadable for this reason. You don't know even if you force yourself, probably need to spend a whole day to read it. And then in the end, you ask yourself, what did I just learn? Well, it's often you can't really tell what you just learned, other than that, there's probably a lot of resources went into generating all those figures. And that is a big problem, and that's the problem that we all have to address, because it's becoming unworkable situation. I think probably 99% of the papers don't really answer a question. So I think the new generation needs to be trained not only in the classical biology, which is another problem that in many countries, in the US in particular, people specialize too early. I think that's a huge mistake. Many students never studied zoology or ecology, botany, evolutionary biology, even physiology. They just started on molecular cell biology. That, I think, is a wrong way of education, that to specialize too early. And my final point is that there are fields like complexity theory, information theory, and control theory, and systems theory. These branches of science, these are not specialized fields. They deal with universals. They're true for thermostat, airplane, and physiology. The concepts are applicable in all of those cases, and they provide this universal language of description. And that's what's really, unfortunately, missing for the most part in experimental biology.
Great. So with this kind of exciting vision of a possible integrative future where we actually understand things without having to read 100 papers a day, or at least try to read 100 papers a day. Because another way of thinking about what you said is it's enough to sample, right? Because ultimately the principles are the same. So if you sample enough and you look at the principles, actually that's good enough. I would really love to, and on behalf of everybody here, first thank you to take your time in this challenging time now to talk to us and discuss your ideas, to dare to generalize and to go out of these details. We really welcome everybody to join us again on 29th of March, where we're going to be hearing Philip Swirsky. And we are looking forward to continue breaking down this barrier between disciplines with you in our virtual seminar series, and to continue interacting with all of you. And again, Ruslan, thank you very much. And let's hope science is a safe haven for us to think about other things and think about.
Yes. Thank you very much. Thank you very much, Carl. Thank you, everybody. Thank you.
Thank you, everybody. Thanks.
Thank you. Bye-bye.
That was good.